we sort of, in this benthic part, we're asking ourselves mainly 4 questions. The one is, how can we quantify the temporal and spatial variations of nitrogen loss we have in sediments, mainly through denitrification and anemox? What can we do to sort of get away from tedious, time-consuming, very spatially limited questions for methods for quantifying benthic exchange that concerns nutrients, metals, and gases, all of them? And then we would like to understand better what are really the regulator for nitrogen and phosphorus release from the sediments, how is this influenced by myofauna, by microfauna, and by macrofauna, and how can we finally sort of contribute to include those measurements into ecosystem models. I'll just present a few data here and just basically run through a few highlights. This is one of the definite highlights we've produced so far, is a seasonal study we've done in the Asker area in towards the Limafjärden, where we've been measuring various microbial nitrogen loss processes, denitrification, anemox, nitrate reduction to ammonium, as well as nitrification. And what we've been seeing very clearly is there's at most stations that are not so much affected by high eutrophication effects, we see a very distinct seasonal cycle with very high rates of loss in August, so in the warmest month of the year. By contrast, what we see actually is that stations that are oxygen limited, or at least temporarily oxygen limited, it is the nitrification process that actually controls the nitrogen loss. So this is actually this red bar that we see here on the right. And this leads us to 3 major conclusions. There's a definite temperature dependence of denitrification. Benthic denitrification is largely limited over the year by nitrification of ammonium, and nitrification as a process itself is not a temperature limited process. It is a very cyclophilic process that seems to operate very efficiently at very low temperatures. And this has large implications for quantifying nitrogen budgets over the year. The other part that we have been doing is, with the bean money, we have built a type of prototype, a benthic lander system that is a very new way of trying to quantify benthic exchange processes. What this system does is an autonomous unit that is placed on the seafloor. It sits there for a number of days. It can be deployed for up to 7 or 10 days. It profiles, it takes CTD profiles in the near sediment bottom, and it does take, at certain time intervals, nutrient samples. At the same time, it measures continuously over this deployment at various levels. So this gives us very, very new data that really there are very, very few data of that type out there for any marine environment. And this allows us to really, at very high resolution, look at what are the nutrient concentrations really right above the seafloor, what are the conditions that the organisms that live in the benthos really experience. And you can see some of these concentrations here. You can see also very steep gradients in oxygen just above the sediment surface. And on the right, you see, for example, a panel of 2 oxygen sensors that are placed at 2 different depths that are monitoring continuously over time. And you can see there's quite a significant variation in concentration over time, over very, very short time scales. It can be a few minutes that suddenly you see hundreds of micromolar drop in oxygen concentrations. So this is really what affects the benthos. This method is then used to calculate fluxes, non-invasive fluxes of nutrients across the sediment surface. And this is non-invasive because we're not just taking the core, we're actually integrating of tens of square meters for the benthic exchange. And these are superior data compared to old benthic flux data. I'll just show you a few more data, multiple CTD profiles we take over these time periods. And what we're starting to do with that is actually to come up with benthic flux maps. This is, for example, a benthic flux map shown for the Asker area, where we sort of plotted now a few numbers. We don't dare to integrate them over the whole period and sort of connect the measurements that we don't have spaces yet. We really want to constrain those with better sediment maps so we can actually get continuous benthic flux maps that we can actually then place these constraints on. The third project is something that Stefano is doing a lot of work on, and this concerns essentially the effects of myofauna and macrofauna. This is published work in MEPS, where we've basically looked at the effects of Marinsaleria on benthic exchange processes, mainly nitrogen cycling, but also metal cycling, gas emissions, oxygen fluxes, and silicate emissions. And it's what we're trying to sort of address our sort of model-based assessments of how invasive species actually catalyze phosphate exchange between the sediment and the bottom water. And some of these results that we have actually created contest some of the model results that have been done previously. And more experiments are needed to do this, longer experiments, but what we see is that the colonization of Marancelera, for example, does not have the much proposed ventilation effect that would actually retain more phosphate in the sediment. It's actually very much the contrast. Marancelera does enhance phosphate release. And the final thing that we have been working on is very experimental work on looking at the coupled metal phosphorus dynamics in sediments by doing experiments by which we shift experiments from oxic environments to anoxic environments, and then basically run relatively long-term experiments in which we quantify manganese and iron release. We do very, very involved speciation analysis of various iron and manganese pools, those that absorb phosphate and those that are coupled via redox cycles. And this allows us to then quantify manganese and iron flux rates from the sediment into the seawater. It also allows us to make inventory calculations for phosphate within the sediment. And these numbers we then can actually use to assess essentially what are the turnover times of reactive phosphorus fractions in the sediments as sediments turn, for example, from oxic conditions to anoxic conditions. Over what time scales is phosphate actually mobilized from these sediments? And one of the surprises that we actually had is when we assessed how long would it take the phosphate, that is the most of the phosphate in oxic sediments is iron oxide sorbed, how long would it actually take for that inventory of phosphate to be completely released? And we come up with numbers of 1,500 days over that time scale. And those are really the first numbers from experiments that allow modelers maybe also to assess when they run their models into anoxia how much phosphate will actually be released. Thanks.